Good afternoon, everyone. Today, I'd like to take you through the fascinating process of mitosis and cytokinesis. These are two critical components of the cell cycle that enables life to thrive. We're going to go ahead and start by recognizing the continuum of phases that occurs during mitosis. I'm going to go ahead and share my screen. So mitosis, which can be divided into several distinct stages. Mitosis is the process by which a single cell divides into two identical daughter cells. It's crucial for growth, repair, and reproduction in living organisms. Mitosis also can be broken down into four primary phases, which are prophase, metaphase, anaphase, and telophase. During prophase, the chromatin condenses to form visible chromosomes, and we can see the formation mitotic apparatus. This apparatus includes the spindle fibers, which are critical for moving chromosomes during the cell division. The condensation of chromosomes actually begins during the G2 phase of the cell cycle. It continues as we process through the prophase. Now we're going to identify a couple of important structures, including in the process. The spindle apparatus consists of microtubules that helps segregate the chromosomes, while asters are star-shaped structures that form around the chromosomes. It's important to note that the nuclear envelope must break down to allow mitosis to proceed, easing the separation of genetic material. Next, as we move into telophase, we witness the chromosomes uncoiling, which is vital for allowing gene expression in the daughter cells. This stage marks the final phase of mitosis. Following telophase, we enter cytokinesis, the phase where the cell divides into two equal halves. Cytokinesis differs in animal as well as plant cells. In animal cells, this process involves the formation of the cleavage furrow, a belt of actin filaments that pinches the cell membrane, ultimately dividing the cell into two separate daughter cells. In contrast, the plant cells undergo cytokinesis by forming a cell plate. The cell plate develops into the vesicles that joins at the center of the dividing cell and eventually becoming the new cell wall separating the two daughter cells. In the plants, the middle lamella forms between the two new cell walls, acting as a stick layer to hold the daughter cells together. Also, it's fascinating to note that in some fungi and protists, mitosis occurs within the nucleus, and cytokinesis is the process that separates the daughter nuclei. The outcome of cytokinesis is two complete daughter cells, each genetically identical to the original cell. This remarkable process not only ensures that genetic material is accurately separated, but also distinguishes between the two methods of cytokinesis in animal and plant cell. In conclusion, understanding the difference of mitosis and cytokinesis reveals the elegance of cellular processes that sustain life, from the condensation of chromosomes and prophase to the formation of the cleavage furrow of cell plates during cytokinesis. These processes are vital for growth, development, and maintenance of all living organisms.